So you gotta get in and. Yeah, mine.
And now you just wired until like 3 or 4. Then I need to like move around.
Oh.
Then I work out after work.
All right, it's still on 59.
The longest flight 59.
One more minute. All right. Thank you. Welcome everyone to our CPC seminar. We are honored to have Dr. Robin Shandas here, who is a distinguished professor of bioengineering at University of Colorado. And I think what's really interesting in reading about your bio is this, like the really bringing things from, I'm going to say bench, but from the basic early research right to the bedside, from not only our kind of ivory tower academic, but also from an entrepreneurial perspective. And what we're really excited to hear about today is his work in, I'm going to say it wrong, I've said everything wrong, Enterotract, which has important implications in health equity, as what I'm understanding, and access for cancer screening. So thank you so much for being here.
I appreciate it. Thank you, and thanks, Jamie, also for inviting me. Yeah, please, it's a small group, so if you have questions or anything, just let me know. Yeah, I've worked on a lot of things over what I've been seeing now, a little over 30 years, and started up many companies and so forth. This is what I'm going to talk about. I feel like it's one of the simplest technologies I've worked on, but also potentially with the most impact as well. And I've learned to make just some qualifications. I'm not a cancer person. I have a 30-year heart, lung, and blood funded program in kind of biomechanics, imaging, heart disease, and so forth. This is a new area that I really got interested in because I started working with a pediatric GI doctor, and then also with Sachin Wani, who's the adult GI doc who runs the esophageal and gastric center of excellence here. And so just learning a lot. But we've made a lot of progress over the last few years. We started up a company. The company actually got accepted into the inaugural Cancer X cohort as a startup company, one of 16 companies in the United States that got accepted. And one of the reasons they accepted us was, as was just mentioned, was because of the potential implications of technology for health equity. And that is something that for me has started becoming much more important and interesting for me to study. As an innovator, an entrepreneur, like the classic model is the better mousetrap model. You build a better mousetrap, you get some doctors to say it's great, you get the FDA to say it's great, and you flip it to a larger company and make some money that way. Like that's like the entrepreneurial dream process. But I've learned that it's not very useful to work on interesting and innovative things if many people can't afford it. So I read an article that says financial toxicity in healthcare affects 137 million people, Americans, and that's just crazy. It's like over half the population, and so part of what I've been trying to just get started on is are there things that we as even academic scientists and entrepreneurs and faculty and innovators can kind of work on this idea. So that's what a lot of this talk is. So some disclosures. Enterotract, as was mentioned, is a company that got started out of this, and then there's another company that's active, Mita, that works on a very different technology. It's actually an orthopedic sports surgical bed for doing hip arthroscopy. It's very different. It got acquired by Stryker, but we're still kind of working with them on that technology. All right, so the string test, I'll pass this around so just so you have a sense of what this looks like. So one of them, this one is like without the little string inside it. It's got a little ball bearing, and then this one is the one with the string. Just to give you a sense of the size of the capsule, it's pretty small kind of capsule. And it was, when Glenn and I first started this company, Glenn Ferruta is my clinical partner, he's a pediatric GI doc, he's the chief of GI Children's Hospital. There was an old version of this string that was available commercially, and that's what this kind of article refers to. It's called the Enro test, and was developed in the 1960s and 1970s, and it was used to diagnose bacterial pathogens in the stomach. So you swallow the string, they pull it out and culture the contents to look for bacteria. So it was used for Giardia, Helicobacter pylori, and so forth. And then some pharmaceutical companies also use that string to collect bile in some of their clinical research studies. Bile is a pretty interesting metabolic fluid. It has certain information about drug metabolism. That's really helpful for pharmaceutical companies kind of going through this. So we did a little survey of literature of groups that had used that original string, and we found actually hundreds, if not thousands, of patients and subjects that had where that original string was used to collect everything from bacteria, parasites, bile, different proteins. It was used in kids and adults. Was left in place for anywhere from 30 minutes to 12 hours. No issues as far as serious adverse effects or complications. So it's very simple but safe device. It's actually considered a non-significant risk device by the FDA. And so what Glenn, that's him in the picture. What he did. He sees these patients with eosinophilic esophagitis, allergy-mediated immune disease that impacts the esophagus, and for those patients, it's sort of like asthma or diabetes. There's no cure for the disease. You got to palliate. You got to monitor, and again, these upper GI diseases. What I'm finding out is that like the kind of serum markers, circulating tumor DNA. Stool markers, saliva, those things don't work really well. They don't manifest early enough in the disease process. They don't have enough sensitivity to be used in a diagnostic screening or monitoring capacity. So for EOE, you have to get a sample from the esophagus. That's the key part. And so Glenn used these devices on his patients. He had 42 patients, kids. He brought them in, he had them swallow the string, sent them home overnight, had them kind of sleep with it, brought them back in the next day, took the string out, sent it to the lab for analysis, and then did a biopsy on them as well. And he published this in Gut and showed like basically equivalence between what the biopsy found and what the string content also found. So he and I and another chemist in Chicago started this company, EnroTrack, a spin out from CU, to kind of commercialize this technology. And one of the reasons we did that is because that old Enro test that I talked about, that company stopped operating about 15 years ago, and we couldn't find any strings. That was the only company in the world that was making those strings. And so we had to create our own company so we could make the strings for some of these kinds of opportunities. So the device is simple. It allows simple, quantitative, and direct sampling of the upper GI system. Like I said, it's a pretty small capsule. We make a variety of sizes of these capsules, so it depends on how far down the GI system you want to go. If you just want to collect esophageal content, we make a tiny version of that. That's like called the Tic Tac size version. It's small. The one that you saw is this medium-sized version. It's used in adults to evaluate both esophageal and gastric samples. And then this larger one, I call that the Mike and Ike's version. That one goes all the way into the duodenum. It goes into the small intestine, and you can collect duodenal bile and other biliary kinds of things. So simple, kind of minimally invasive and location-specific sampling. We changed the design of the original string a little bit to kind of protect the samples even more as you pull the string out. You can do office-based administration. You don't need specialized facilities. You can do it in a community clinic, those kinds of things. And the idea is that you collect the sample sent to a reference lab, the reference lab analyzes it for different markers. So far, the string has been used in about 800 patients. There are 9 clinical trials. Ed Liu at Children's, who runs the CDAC Center, is just starting a trial in CDAC with this to look at the ordinal cytokines with the string as well. So the method is pretty simple. This is one of my PhD students kind of swallowing the string at home. She runs a microbiome project, and her idea is to send these things to people's homes so they can swallow it. So yeah, you just swallow the string, and then you have somebody hopefully that likes you pull it out. And so that's it. It's a very simple kind of process. It's the string that's the magic here, right? It's not the capsule. It's nothing that's just the string kind of sits in there, and sort of it has this very absorbent kind of core. And so if you have an early malignancy, for say a secreting tumor or something like that, the string is sort of in the right place at the right time, because you can do it, every few weeks, every few months, however the disease kind of process manifests. So these markers are picked up by the string, sort of sits in the mucosal layer, and then we have these fibers that kind of stick out the side of the string, called fiber lock. That when you pull the string out, these fibers kind of align against the direction of pull, and sort of reinforces the contents of the string, kind of protects them from that brief period when you're pulling them out. So that has worked pretty well. So I mean, there's been a lot of studies that have looked at this. This is a summary of a phase 2 multi-center trial, 5-center trial in adults and kids, all with EOE. And so out of the 344, I mean, the primary kind of issue, yeah, please.
Oh, sorry, just a quick question.
Yeah.
So there's nothing in the capsule that like opens and expands and that pulls up the sample, it's just on the string?
Yes. So there's no sponge, there's no
Yeah.
balloon, there's none of those, like those other methods that
Yeah.
are there.
Okay.
Yeah, nothing like that. It's the string that's the secret. And so what's interesting, so some of those other methods, they're like specifically set up to like scrape the epithelial lining and gather cells, right? So I wouldn't call this a cytology device, although it does pick up cells. We've done some histopathology and showed that we do see cells, mucosal cells that enter the string, and bacteria and other things. But it's more like it samples the mucosal layer.
Okay.
Is what it does.
So, and on the mucosal layer, what kind of data, and you mentioned some so far, what kind of data in general, like I'm thinking psychologists would use cytokines or inflammatory markers and like would that?
Yeah,
Same as
all of that, yeah.
Yeah.
So it picks up a lot of things. That's one of the things we realized, because the original kind of Glenn's original vision was going to pick up some inflammatory proteins that correlate with eosinophilic disease. And it does, it picks up those, but we're also realizing that it picks up a whole bunch of other things as well. And so, and then I'll go through some of that as well. But yeah, I mean, the mucosal layer, again, I knew nothing about the mucosal layer besides a class in graduate school. It's a pretty interesting layer. I mean, it's like the first line of defense, to the environment for your pulmonary and GI system. So it's sort of like this, like heavily biochemically active kind of interface, right, between the immune system and the outside environment. So there's a lot of things that happen there, and if you can sample it, this idea of, mucosal immunity is a kind of big field. So this idea of like being able to evaluate that easily is somewhere that we could kind of eventually go here.
Yeah, oh, that's great.
We did some surveys, and it's not surprising, for parents who have kids with EOE or people with EOE, if you kind of compare this to endoscopic biopsy, they vastly prefer this method. And we've shown, like, again, pretty extensively that it picks up not only similar things as endoscopic biopsy, but at least for EOE monitoring, that test has similar sensitivity and specificity to endoscopic biopsy. There's some data that Sean Schroeder from Phoenix presented at the last DDW meeting. This is from 67 kids, and it's actually now, she's going to publish the first year from a little over 100 kids. And so this shows that, I mean, basically, these are kids with EOE, so obviously they have issues with dysphagia and other things. So most of them have, more than half have no issues. About a third of them kind of have some gagging as you swallow it. And about 3%, a little less than 3%, they're unable to swallow it. And she gives them kind of 2 tries and then it's off to the endoscopic suite. Now, when I looked at this compared to like regular pharmaceutical capsules, it's actually very similar. 40% of patients have challenges swallowing pharmaceutical capsules, so it's not any different than that. So we started looking at EOE as our first kind of product, essentially. Impacts about 500,000, requires frequent monitoring, like I said, to control the disease. And this is one of the diseases where clinical symptoms do not reflect disease severity. So you may have the disease, everything seems to go fine, and then suddenly you eat something, the food triggers the esophageal inflammation, gets stuck in the esophagus, you gotta go to the emergency room. And so you need kind of other ways to monitor the disease. And again, it's one of those areas where, there have been several studies looking at blood markers, saliva and other markers, and they don't work very well in this disease. You need a sample from the esophagus. Endoscopies, obviously, it's useful for diagnosis, and we don't claim that we are trying to diagnose a disease. We're using it for screening and monitoring, and so it's a challenge. And then the other thing that even though my clinical colleague is a GI doc, one of the things I realized is that the allergists and immunologists are the ones that have expertise in this disease, and they're the ones who see it, but they refer to the GI doc, and they usually lose the patient at that time. And so because again, there are no tests to evaluate it. It's not like an IG mediated kind of, you do your skin prick testing and things like that. And so we've had a lot of response from allergists around the country for this test, because it allows them to keep the patient, monitor them, maybe even make some money. And so this is some data that was published very recently from a 1-year experience here in Denver, like looking at a few case studies. So here's a patient with EOE. He's had about six biopsies in the last 6 years. Seemingly her EOE is well contained, but really it's well contained because she's avoiding a whole bunch of foods. And so in order to identify which of those foods are actually the triggering foods, that she'd have to go through multiple kind of endoscopic biopsies, and certainly has pretty poor quality of life as a function of this. So they went ahead and used the string test here. So it's a little hard to see, but they started doing a food kind of challenge. So they added corn and they did the string test. And you get, so we report this EOE score, and it's a probability of disease kind of score. If it's greater than 0.53, that means the patient has active disease. And so here it was 0.414. Patient, the corn is not triggering the disease. They kept the corn. They started adding wheat on there, and you can see the EOE score jumps up. So it goes to active disease. So this patient, it's the wheat. That's the challenge. They remove the wheat. Added dairy, the EOE score goes back down. So it's a very quick and convenient way of assessing immediately what's happening for that particular patient. This, if you had gone through endoscopy, would have taken like at least 2 years to go through the whole process. They did it in about 3 months. So the patient's foods have been quickly identified. There's no need for those additional biopsies, and quality of life has improved. So certainly there's a time savings, there's also cost savings, across the board. This is like for the payer, the caregiver, the patient, the health system, there's sort of cost savings across the board. And the paper kind of goes on to talk about other case studies where this one, they were trying to titrate disease treatment, right? The patient was on PPIs, kind of wanted to figure out what the level of PPIs was best to control that patient's disease at that particular time, was able to do that quickly. And this was sort of combination therapy where they wanted to see the patient wanted to eat more types of food, but was having the triggering symptoms, and so they were able to titrate the therapy as well to be able to do that. So I like the concluding sentence in this. So the ESD led to management changes and was safe, well tolerated. And it was quicker and more affordable than endoscopy. Again, this is the first set. This was about 38 patients, I think. There are a few more studies that are coming out, so we'll see where it's going. But from a kind of, business perspective and moving this forward, as I said, it's starting to get pretty popular around the country. We just hired like our first 2 salespeople, and there are a variety of kind of clinics around the country that are starting to use it. So that's kind of pushing forward, but kind of, going to your question about what else can this thing do. We noticed that it takes up DNA as well, and so this is where the conversation with Dr. Wani kind of was very revealing for me, because he specializes in esophageal adenocarcinoma and trying to ablate, catch it early and do endoscopic ablation on them. And so, we looked at, can this test be useful for that to prevent early deaths and decrease financial toxicity. A huge number of people have risk factors with this, but obviously they're not going to go through endoscopic screening for this. And same kind of thing, you need a sample from the esophagus, for this. And in fact, if you look, there are clinical guidelines for endoscopic screening for esophageal adenocarcinoma, and they've been around for over 2 decades, but less than 10% of people actually kind of go through that. So that's what we thought, we can start making a difference. So this idea of like simple home-based sampling, sort of like like the Cologuard, for colorectal cancer, where you send the kit home. And so this same idea, can you send this string home to somebody, they self-administer or have somebody else administer it, send it to our lab, and we do this. So that's that combination. I mean, there are lots of great technologies on this graph here, but that combination of like being able to do it at home, you don't need an endoscopist, you don't need specialized facilities, so forth. It seemed like we kind of captured that part of it. So I've shown that it captures kind of similar markers as endoscopic biopsy. There have been other studies, talk about, like, for instance, the microbiome. There was an article published that showed that for the esophageal microbiome, the string test captured both similar amounts of bacteria as endoscopic biopsy and similar types of bacteria as well. And I'm happy to send any of these papers to anybody. And then we have, Kat, my PhD student, working with Emily, who's over there, and others, we're doing a study where we collect kind of location-specific different biomarkers in the upper GI system. So for this one, we use that longer version of the string that goes all the way to the small intestine. And the idea is the volunteer swallows it, we leave it in there for a couple hours, and then we take kind of 2 centimeter sections from the esophagus. We take 3 of those, 3 from the stomach, 3 from the duodenum, 3 from the jejunum, and then we do different kinds of analysis on that. So we started getting some microbiome data. So that's kind of the load data from the string in the different 2 centimeter segments. We can get proteomics. We're running proteomics panels on these, and we're also can get DNA to do DNA methylation as well. And so it seems like it collects a lot of these markers. Now the question is, and hopefully you all can help me with this, what do we do with all this information? It's easy to get. You can send it out anywhere. What do we do? And the idea, again, this is kind of the disease screening kind of idea, is that it's easy to administer, right? So for diseases sort of nucleating locally, and it's starting to produce those early markers. I mean, can the string kind of be there at the right place, right time type of thing, pick up those markers, and then get a sense for that early kind of stage. And so the idea for home-based screening is, you obtain the sample at home, you ship it to our lab, we do the analysis, and we send you a report on that. So Sachin, was excited about this. So we started a pilot study through the Cancer Center, through his center, to look at patients with Barrett's esophagus and esophageal adenocarcinoma. And the idea is that the string would be administered either within a short period of time of endoscopy or within 3 months of a confirmed endoscopy with confirmed kind of diagnosis. String is collected after 1 hour, and this is actually an older slide. So it says 29, but it's actually, we've collected 80 patients so far. The pilot study is actually done, so we're starting analyze the data. But again, we can collect not only the DNA, but we can also look at microbiome, proteins, and other things as well. So there's a huge amount of data that this is going to generate. In fact, that's going well. The patients tolerate it great. In fact, talking about like the balloon and stuff, I was talking to one patient who did undergo the balloon, and he preferred this. So that was anecdotal, but still, I was pleased to hear that. So this one, we're analyzing the 80 subjects. We're submitting a revision to the protocol to do another 400 patients with this. And so, so that's sort of pushing forward with that study. And then another area, so a group at the NIH approached me from the National Cancer Institute, and they were interested in using the string for detecting Helicobacter pylori, which is, as you probably know, a bacterial infection. It impacts over 50% of the world's population, right? And it's an infection that under certain, kind of constraints of virulence and phenotype and genotype goes on to create ulcers, gastritis, and for a small percentage of those gastric cancer, as well. And even though environmental and other factors are all correlates to the development of gastric cancer, H. pylori is still one of the key kind of determining things. So there are lots of studies. Again, I'm very much a novice at this. I'm still learning a lot about this, but there's a lot of studies that are showing that Helicobacter is an old bacteria, but it's changing, and it's changing in both in response to lots of antibiotic treatments, and so some strains are becoming antibiotic resistant, but also there are differences based on where in the world you are. So, for instance, if you are from say South Asia and you have H. pylori, the chances that you get gastric cancer is a lot less than if you are from China or Japan, for instance. So there are these interesting kind of phenotypes in H. pylori. So the idea that it's not just that you need to detect whether H. pylori is present, but you need to identify the kind of H. pylori it is. So things like stool tests are good, right? That's the standard for evaluating whether somebody has H. pylori. There are some markers as well, but those don't give you the type of H. pylori that's actually present in your stomach. You need the gastric biopsy for that. So as I said, the serum, stool, saliva tests have limitations with this, and so so we were asked to see, well, can we work with them on this? So I did some background research on this, and remember that first slide I put up there where we talked about how that old string test was used in this. It's actually was used a lot for looking at H. pylori, and so there are lots of studies, and I've started talking to many of these groups where they looked at string versus biopsy in a lot of things using culture, PCR, a variety of things, and showed excellent kind of specificity and reasonable sensitivity. I mean, outstanding specificity for the test, reasonable kind of sensitivity as well. So we are creating kind of our own version of this, and the idea is that we will combine kind of the collection of gastric samples, and that's just with the standard string test. You can collect it, you can do some pH kind of evaluations of the string, but the idea is that we'll take sections from the stomach, and there is a urease kind of test, a pH kind of test where, so again, Helicobacter is a very interesting bacteria. It produces urease that converts kind of uric acid to ammonia, and it sort of decreases the local acidity, so it increases pH locally, and it's a survival mechanism for the bacteria. And so this test is a way to detect Helicobacter by detecting the presence of the enzyme urease and its conversion to ammonia. So we just do a simple, basically litmus test. So we take the sample, we create a little custom urea broth with phenol red in there. Again, very simple, and we go ahead and put the sample in there. If it turns red, that means the bacteria is present. If it stays yellow, it doesn't. You can do that within 30 minutes, like right there on site. So you don't need any specialized kind of facilities. But the other aspect is that we can also like store more sections from the gastric region. There are these universal or like preservatives you can buy now that allow you to store these bacteria at room temperature conditions for longer periods of time until you can go and get a PCR or some kind of phenotyping process done. So it allows you to store these samples for that subsequent phenotyping that helps with kind of identification of virulence and phenotypes. So when I proposed this, I mean, people got really excited. So actually, now we have like a large number of studies that are starting up around the world with this. So there's a group in Mali that's going through the NIH, and they're very interested in not just like people in Mali, but people from many of those regions that have emigrated to the United States and how kind of things change as that happens. There's this African Helicobacter Genome Project, a huge project across multiple countries that's starting to use our string. There's a group in Latin America. Javier Torres is somebody that spent like 20 years working with the old string. He was super excited when I told him we have the string again for this. And then there's a group in India out of Kolkata, but they have like a very large kind of set of studies around the country as well on this. So we're starting to send strings to them for them to try out. They're kind of giving us feedback on how it works and whether there are things that we need to change and so forth. So I'll end with a couple of kind of other areas that I feel there's some interesting applications. And so, as I said, the string collects kind of duodenal bile as well. And so I got a call from a scientist at GlaxoSmithKline, and he was one of the guys that's published. Remember that one of the first papers I put up there on the old string. He uses the string for their like ADME studies, their absorption, distribution, metabolism, excretion studies. Even though and so you comprehensively profile the subject there. You get blood, you get stool, you get urine. But bile was one of those things that he wanted, but it was very difficult to get. Endoscopically, there is a method to do it, but there's no way you're going to get volunteers to volunteer for that method. So he got excited that we had the string again, and so they ended up using many of our strings in their studies. So it's used in the ADME studies. It's used in kind of microdosing, microtracing kinds of studies. As well. And so it's that idea where you have a drug, it doesn't matter what drug it is, but you go from kind of the healthy volunteers to kind of the not so healthy and to the patients, and that kind of process, kind of trying to get healthy people to volunteer for this. The easier you can make it for them, the more success you'll have. And so the string kind of allows them to do that. And so this is a paper they published last year about their first experience with the string for certain drugs that they were looking at and combining with sort of a microtracing type of study as well. So once they got started, I actually got calls from ICON, which is a CRO, LabCorp, another CRO, obviously Charles River, and then we just started a big project with Bayer in Germany as well that's doing this. So it can collect biliary kind of information, and so there's either that generates any ideas. I'd be happy to kind of talk with you about that. This is sort of how Dr. Liu at the Celiac Center kind of got interested. Again, same kind of issue. If you're doing gluten challenges, kind of using serum kind of cytokines may not be as sensitive to actually getting the samples from the place where those are produced, where the inflammation is happening. And so that project just got kicked off as well. And then some other studies, I mentioned them, esophageal microbiome. There are 3 or 4 papers that have been published kind of documenting how useful the string is compared to biopsy, and also looking at it for things like EOE as well. and for patients with cystic fibrosis. And this one is sort of more to evaluate IV therapy in patients with cystic fibrosis, and to see whether the microbiome may be a sensitive marker. And I'll say it's an incredibly sensitive marker. I would say it's almost too sensitive in a sense. And then I'm glad Emily's here because I want to shout out to her because she has now, she's been using the studies, the string in her nutrition studies as well. So this is work that they presented based on the nutrition protocol that they have going on. And the idea here is, one of the things that they were finding is that the stool microbiome is not necessarily your gut microbiome, right? So this idea that, everybody, when you talk about gut microbiome, it's usually stool that you look at. I'm sort of, I don't think that's correct. And so they did some studies where they looked at stool for 6 volunteers in that study, and they looked at small intestinal microbiome using the string. And again, very different in terms of kind of content and composition, including if you look at the ordination charts. Again, there are kind of clear differences in there. So again, it seems like there's something here, and certainly I think I've had some spirited discussions with Emily about what to do with this stuff. Like what do we do? But then she told me that, she just got a grant to look at cancer survivorship, that she would like to use this you add the string as an additional kind of metric to look at that. So there may be some opportunities there that certainly could be talked about. We're also working with cancer. With Cancer X, they've put me in touch with the group at Moffitt Cancer Center on doing this, and also a group at Atrium Health as well, that are interested in kind of remote monitoring of nutritional status in some of the patients as well. So so that's sort of that's what I have. Thank you. I'm happy to take any questions, or more interestingly, kind of talk about where it's going to be used. Thank you.
Couple of questions, Roman. First of all, this is incredibly intriguing. What has to be done with this string in certain things? I think you alluded to some things that you do to preserve some things, but in terms of the process of the string, can you send this out into remote areas and have it sent back in bulk, or those kinds of things, and expect it to be stable? And
I think it depends on what you want to measure.
Okay.
So some of it, there has been work done. There's a group in Australia that used the old string, and I can send you that paper as well, where they wanted to look at Helicobacter, and so they came up with a preservation buffer where you don't need to refrigerate it or anything like that, and kind of send it out to labs. The proteins that we look at for EOE right now, those are frozen and sent to our lab, but they're frozen more because that was what the validation protocol had. I've looked at those proteins. They're not, I don't see any kind of sense that they're thermodynamically labile. And so I don't think that there's issues. Proteases are an issue, and but if we ship them overnight in controlled conditions, we should be okay. So we're actually testing that right now. So the short answer is we don't know, and we kind of want to benefit from the experience that you or others may have in whatever biomarker you're trying to study, in what the whether we can come up with a preservation buffer or something to store. The microbiome right now is just frozen right after it goes into -80.
So another question. Now, are you using the string from the previous company, or have you made some additions or modifications to the string in terms of?
We've made the mod. So we have 5 patents on the string based on the modifications. So that the fibers that come out, and there's some kind of things about manufacturing it. The way the distal part of the string, the old one kind of was fused to the capsule, which is not not an elegant kind of design. So we changed some of those things yeah, so we have some unique kind of aspects to it.
I was really interested in the PK work and how that, I mean, do you see this as a shift? Like, what are the other advantages? I mean, you could, to someone who doesn't know anything about, I know very little about PK modeling, but thinking about that over like serum samples.
Yeah, I mean, again, I don't know much about it either. But I've been told by the scientists at Bayer and GlaxoSmithKline is that, this doesn't mean that you do away with serum testing or serum sampling or something. So the bile is just an interesting fluid that they want to get their hands on. It just appears like temporarily and it goes away. It's like you don't see it downstream the GI system. So you need to like be there at the right time, kind of right place to capture it. And it has these particular metabolites that seem to be very useful. So, yeah, I mean, there's a lot of sophisticated spectroscopy in those studies that I don't fully understand. So I'm happy to send those to you. But they seem to be pretty excited. I mean, Bayer is very excited. They have a big trial that we just got this cleared in Europe to be used for all their clinical research studies. So they'll probably publish another paper on this as well. So it's an additional thing. And I'll say one of the caveats is, it still doesn't seem to be a quantitative marker yet. It's sort of like a semi-quantitative kind of information. But the idea is that because it provides this additional information, maybe it's like a screen that can be used kind of early in that process.
Yeah.
Sorry.
Go ahead.
Just, this is so fascinating, and I do work with Sachin, and I must have missed that the string can collect all those other interesting things, so I'd be very curious if you had a list of what kind of biomarkers your string can be assayed for or analyzed for. As a psychologist, I typically, in the past, have looked at salivary biomarkers for cortisol or, serum plasma, just inflammatory biomarkers, and I'm wondering, just with the microbiome piece, I'm working with metastatic colorectal cancer patients right now with partners. And has there been any data to your knowledge that have looked at like dyadic associations with the markers that are gathered between patients and their partners, for example, or associations with psychosocial outcomes?
Not that I know of. I mean, so one of my students approached me, he's like, there's this relationship between the gut and the brain, right? And
Yeah, sure.
The gut and mental health. And so he was very excited about that. So yeah, I think that depending on the intervention you want to do, this is a simple kind of product to try out. So we're working with starting to work with the diabetes group, Nita Razzulli and Jane Roosh and those guys who work at the VA. And they're obviously very interested in metabolic function and kind of markers of that. And so can the string kind of allow, certainly discernment between, people with kind of deteriorating metabolic function. But there could be other interventions, absolutely, as a function of that. I think it's wide open. I think one of the interesting things I've seen is, there've been 3 grants that have been submitted through Pediatrics Children's Hospital that use the string, and all of them have gotten accepted. There's one at the NIH, one of the ones I put up to look at this screening between that group in Mali and kind of immigrants from Mali here that got funded. And there's another screening, one large one between North Carolina and Virginia that also looks like it's going to get funded. So it seems like there's a good opportunity for it to be positioned as an innovative, method. But it's something that, we're happy to like provide the string and kind of provide that information. But I know nothing about that, so I'll probably ask.
No, it's just such a great product, because one of the things we struggle with in the field is getting biosamples from partners as well as patients, because patients come into clinic and you can get blood samples right then, but now we're missing this kind of other piece. At home salivary testing is burdensome over several days and things like that, so I really feel like this could really unique niche. And could-
Yeah, so the Helicobacter, they have some similar issues because so if one family member is infected with Helicobacter, it spreads to the others. And so identifying that early, is a big push. Like in Korea, they have a huge like public health outreach program where social workers go to people's houses and talk about testing and so forth. So that's where, like catching this early, because stool testing is still, still there's a delay between when you might be able to catch it, same with serum testing. So they're thinking this may be an opportunity to catch that early infection as well. But what's good is they're saying that then they can have the other family members also try, use it as well.
Yeah.
Are there things that you can't detect with this that you'd like to be able to?
Well, I mean, I know Emily's interested in RNA and all the things, and we're interested in RNA on the cancer side. So one of my PhD students is looking at that, but I think that's a challenge. We have to figure out the buffer and all of those kinds of things. I don't know. I think you did some initial analysis, right? Or
Yeah,
something
Kartik
like
had
that.
taken some of the sample strings from when you guys did some internal testing, and he had enough load to be able to detect, but I don't know.
Okay.
We haven't looked at it with any of our mini med samples.
Okay. So yeah, so the RNA might be one thing. So we are doing full proteomics, and I can share the proteomics data with you. We pick up about 600 proteins with the string. One of my other students is looking at that for duodenal kind of small intestinal thing. She said that she was picking up clearly kind of small intestinal proteins from the string. So we have that data. I'm happy to share that as well.
Anybody online with questions, Ryan?
No, it's just me.
Okay. All right. Well, thank you very much.
Thank you so much.